To introduce myself, my name is Thor Rollins. I am the Senior Director of our Toxicology and Extractable Leachable Group at Nelson Labs. I've been at Nelson Labs for 20 years, and 18 of those 20 years I've been involved in the ISO 10993 working group, the TC 194, Technical Committee 194. So we're responsible for writing the ISO 10993 series, which some of you may not like me now because you know that I had my hand in that series, but hopefully, we can help a little bit with understanding where we're at and where we're going with that. There's a couple of things I want to make everybody aware. So today we're going to be talking about some of the problems we're seeing right now with turnaround times with biocompatibility. I think everybody, if you've done biocomp tests in the last 2 years, have realized that we have some issues right now with turnaround times. I want everybody to understand that at Nelson, we actually don't do any of the animal tests. We provide them through subcontracting. We've dedicated ourselves to in vitro alternatives, but we do provide a lot of subcontracting effort. So that allows us to use multiple labs throughout the industry. The information I provide today is not on one single lab. It's more of a general kind of issue that we're seeing in the industry. So don't think I'm making fun of one lab or another and things like that. I really am talking about the general problems we're seeing today and what are some tricks and tips to try to get your product through the FDA quicker with the constraints we have right now in the industry. So that's what the topic is today. And if you have any questions, I'm more than happy to answer them. We only have 45 minutes today, but my booth is, I wrote it down so I could look, 3135. So if you want to come afterwards to the booth, I'll be there all day long and we can chat about anything biocompatibility you want. Okay, so the first thing I want to make sure everyone understands is the new changes that we made to ISO 10993 Part 1 in 2018. Just so that everyone's aware, we're actually changing this again. We can't keep well enough alone. So -1 actually is in a draft right now, and we're updating that draft as we speak. If you want to know more about the new changes that are coming with 10993-1, tomorrow morning at 8:30, I know that's early, but 8:30 we're doing a conference, it's free upstairs, and I'm talking about what changes are coming to -1. So if you guys want to come and listen about what's coming down the pipeline in 10993-1, then I'll be talking about that tomorrow morning at 8:30 upstairs in the conference, and it is free. So anyway, so this is the new chart of 10993-1. Okay, and there's a couple of updates that I'm going to cover really quickly because it helps you understand how we can maybe save time with evaluation of biocompatibility in today's market. So the first changes that we made is we added a column. And this is important to understand. If you're not aware, this column is material or physical and or chemical information. So this is the first time we've added a column to the chart since it was first put into the standard back in the 90s. And as you can see, it's the only column that has an X to it. So what that means in our conversation today is this is the only thing that you guys are required to do per the standard. Everything else, as you can see, is an E, which stands for evaluate. Too many times, we're just used to, in this market, if it has something marked in that column, we do the testing, right? You call up your lab and you say, I need to do sensitization, irritation, acute systemic toxicity, whatever it is, and we do the testing. And that's how it's been for my 20 years of my career. Unfortunately, right now, and we'll show this, the turnaround times of these tests are starting to get problematic when it starts to actually doing biocomp, completely honest. So it might be time for us to start evaluating instead of testing. And believe it or not, it does work. We're very successful with doing evaluations depending on the quality of your physical and or chemical information. So the opportunity for you to evaluate your endpoint depends on the quality of the material information you have up front. Those are important. It's almost like a seesaw or measuring table. The more you have on one, the better your evaluations can be on the other, okay? There's a couple of updates here that I'm gonna spill out. I'm not gonna spend as much time because it's not as important as that material, physical and chemical information. We made a mistake. We left off sensitization. That gap there is supposed to be sensitization, so keep that in mind. We added material-mediated pyrogenicity. It was always there, but we actually put it in the standard now. We separated subacute and subchronic toxicity, so now we have two separate instead of together. We added carcinogenicity. Just a little side note with carcinogenicity. If you have an X on that mark, you have to evaluate carcinogenicity. There are three ways to evaluate carcinogenicity. To have such good material information that you know your carcinogenicity risk is low. Secondly, is to do extractables and leachables and evaluate it with a toxicological risk assessment. Or third is to perform a carcinogenicity animal test. And that animal test is about 1.5 to 2 years long, and it's about $1 million to $2 million. So not very many people have done the animal test, as you can imagine. But historically, before this standard came out, a lot of people would just say, oh, my genotoxicity is good. I don't have to worry about carcinogenicity. But now, because we have it on this with an evaluate, the FDA is requiring you to evaluate it if you have it there. Just a little side note. We also added reproductive toxicology and degradation. Just to let you guys kind of know why this is important. We don't have any E's on that, but if your device, if you think it's gonna degrade or have a possibility to degrade, or if you're gonna use it in an intended population that is reproducing, like pregnant mothers, for example, then we have to worry about those endpoints. So that's why we don't have an E there. But for those of you that are joining new, I just wanna make it home here. So we're trying to justify out of biocompatibility testing without doing it because of timelines. And the most important thing about doing that is understanding your physical and chemical information. So that's why I have this up here. Also, for the new people, if you want a copy of these slides, just give me your business card afterwards. I'll give you the slides there's nothing on here that I don't care about giving away. Okay, so to kind of hit home that point, down here at the bottom, as you can see, X means you have to do it as part of the risk assessment. Remember, that's only the chemical and physical information gathering exercise. Everything else is endpoints to be evaluated in the risk management process, okay? Not automatically test. And that's an important difference. Okay, so let's talk about why this is impactful today. So up here, and once again, this is not a specific test lab. This is throughout the industry today. We subcontract to all the major test labs, so we kind of know the turnaround times, okay? So this isn't picking on one particular test lab. But up here, this was historically the turnaround times we saw with biocompatibility pre-COVID, okay? So this was very normal in my 20 years career. Didn't change a whole lot. Today, this is what we're seeing. So for material mediated pyrogen, we went from 4 weeks to 7 to 8 weeks today. For subacute subchronic, 6 to 8 weeks to 10 to 13 weeks. For irritation, 4 weeks to 7 to 8. Sensitization, 8 weeks to 13 to 15 weeks. That, by the way, that kills me, right? Because everybody has to do sensitization. So that means even if you just did the big three, cytotox, sensitization, irritation, your turnaround time was 8 weeks, and now 13 to 15 weeks to get that test done. The next one is acute systemic, went from 4 to 7. So these are your new turnaround times today. Now they're gonna improve. There's a lot of obviously constraints that we're seeing in the COVID times with personnel and animals and things like that, but it's not getting any better quickly, okay? We still see these issues. So your choice is to either put up with these turnaround times or be creative on how you evaluate biocompatibility. No longer checkbox these tests, but look at ways to identify or evaluate these endpoints through a risk assessment. And believe it or not, the FDA and notified bodies in China and Japan all take risk assessments if done correctly. It just takes more work up front. It's easy just to submit the test request form and get the results. But you gotta weigh that now against the turnaround time we're seeing, okay? Okay, so we're gonna look a little bit about alternatives. Now remember, these are mostly animal tests that we're looking these turnaround times at, right? There are in vitro alternatives that are being generated right now that we can use that are quicker than the current animal tests, okay? So we wanna look through risk assessments through a biological evaluation plan as one of our options. We wanna look at in vitro irritation as a second option, and we'll talk about what that looks like and where you can use it and when. Extractables and leachables. Please use extractable leachables the best you can. In fact, we have a real life example of actually here in California, there was a bare metal stent company, and the owner of the stent company was adamantly against doing animal tests. This was pre-COVID, so it wasn't turnaround time driven, it was just her morals, right? She didn't want to do any animal tests. We're able to get that stent, permanent implant, approved through the FDA without a single animal test. That includes implantation, that includes sensitization, that includes subacute, subchronic. So it's possible, it does take work. It's not easier than the animal test, but we can do it, and it can end up being quicker and cheaper than the animal test. Extractables and leachables, like I mentioned, and then the sensitization evaluation, which becomes maybe the most important one. So like I said, that's one that everyone has to do, and we've seen these long timelines. So let's go really quickly through those options. The first one is doing a really good biological evaluation plan, or BEP. I'm going to talk a little bit about what a BEP is, right? So everyone knows. This chart has been in ISO 10993 for as long as I've been doing biocomp. It's not new. It's a flow chart for helping you identify the risks for your device from a biocompatibility perspective. It's a great place to start if you're new to BEPs. These are questions that you can help and ask yourselves, ask your company to try to gather the information needed. If you need help writing a BEP, we do it every day. We do 3 or 4 every day, but I'm happy for you guys to try yourselves. I want to develop more and more biocomp experts. We don't have enough. So I want all of you to become biocomp experts in the next 5 or 6 years. So go through this flow chart, learn how to do it. These are the questions that are important. The first step, though, in this is still to obtain device material information identification. Like I said, it's the most important part that people forget, because it's easy just to get the testing done and you don't have to do this part. So what is physical chemical information and material characterization? There's some confusion in the industry about which is which. Up here, we have the umbrella one, which is physical and chemical information. There's lots of things that go into physical and chemical information. A lot of people confuse this with extractables and leachables. Extractables and leachables is part of a physical and chemical information gathering exercise, but it's not only the choice. You have other things like material, like from suppliers, information from suppliers. You have their biocompatibility information. You have physical characteristics, all these different parts of your physical and chemical profile. You also have material characterization, which once again is not only extractables and leachables, but this also goes into things that helps characterize your materials, like how rough it is, how smooth it is, or its flexibility, its leachable profile, things like that. So there are multiple ways to get supplier information. Like I said, this takes a little bit of effort on your end up front, but it could save you time and money on the back end. The first thing is to get information from your suppliers. Be careful on what information you get from your suppliers, and we'll talk about it in a minute. Not all information from your suppliers is judged the same. There's also predicate testing. The big companies do this the best, and it makes me a little frustrated that the big companies do this because they have the resources, and so they save time and money, and all the small companies end up just doing all the testing because they don't have those. But the big companies develop databases of biocompatibility information, right? So they have database of materials and processes that when they are making a new device, they can go in and pull that information from their own database. And now they're not doing as many or any biocompatibility tests because they already have that information from previously tested medical devices. This is a great way to reduce the amount of testing you have to do. It does take upfront work to develop and maintain, but it saves you in a long run. Also, extractables and leachables or material characterization can help fill up any gaps. So this is a soapbox for me, so just give me a second to step up on my soapbox. We do a ton of BEPs at Nelson Labs, and we ask for supplier information, and I can't tell you how many times we get ISO 10993 compliant, or biocompatible, or medical grade, right? That means nothing. ISO 10993, there's no standard that says what is ISO 10993 compliant. There's no standard for medical grade, okay? So when you get this from the supplier, don't just take it and say, oh, it's medical grade. Ask, what did you do? Can I see the reports? When were the reports done? Were they done 20 years ago? Are they still relevant to today? You need to dig a little bit deeper because now you're relying more on this information to generate your own risk assessments. So now I know this is sometimes difficult to get this from your suppliers, but this is one way that we can help justify out of biocompatibility tests with more and more information from your suppliers. I would recommend talking to your suppliers beforehand to make sure they have this information on file to be able to give to you before you start going down the testing route. USP Class 6 does have testing associated to it. So if you want to be USP Class 6, as a raw material, you send it into the lab and they do testing. They do these 3 tests, which is irritation, acute systemic toxicity, and a 7-day implant. Now, I love USP Class 6. I think now it's not really made for raw materials and medical devices. It was for containers for pharmaceuticals, but it's still some good information. Just be careful because one, that implantation is a 7-day implant without any histopathology. So I can't really draw any information from that implantation test. So that doesn't really help me from a biocompatibility standpoint. If you send it to some labs, they'll do a combination ISO 10993 USP Class 6 test regimen that will do better implantation data that we can use. But if you just do a plain USP, that implant really doesn't help us from an ISO 10993 compliant standpoint. The other thing that I sometimes run across is USP Class 6 does not include a cytotoxicity test. Cytotoxicity is by far the most sensitive test we run. At Nelson Labs, we did a 10-year investigation, and we looked through all the failures for biocompatibility. 93% of the time, if you're going to fail, it's going to be cytotox. So if you're going to run a USP Class 6, or if you're going to accept a USP Class 6, make sure you do or they have a cytotoxicity test. Because if not, it doesn't really mean as much without that screening test with it. So make sure you see cytotoxicity along with USP Class 6. Okay, so that's your material gathering exercise. I don't have time to go into more and more details, but if you guys want to get into the nitty-gritty about how doing a biological evaluation plan, I love talking about this stuff, as you probably can tell. So come and talk to me at the booth and we can have a good time. I do want to mention an in vitro test that's now in the standard that we can look at doing that's quicker than the animal test is, which is in vitro irritation. Now we recently separated ISO 10993-10. So -10 was for sensitization and irritation. We now have -10 just for sensitization, and irritation is now -23, okay? And the main test method in that standard is this in vitro alternative. So no longer is the animal test the standard in the standard. That's kind of weird to say. But the standard test in 10993-23 is the in vitro. So let me tell you how it's done really quickly. It's very similar to the animal test. So we take your device and we still extract it just like we'd do with a rabbit, okay? Polar, non-polar, using 50 degrees C for 72 hours, very similar to the rabbit test. But so we extract it, then we expose that extract to tissue. So not to an animal, but we use tissue. And what we use is reconstructed human epidermis. And if you want to just learn how to say that, you can impress your friends, right? So we shorten that to RHE. But reconstructed human epidermis is that. It's actually human skin that we produce from stem cells. And it looks just like our skin. In fact, it has little fingerprints and everything. So we can extract these medical devices and expose this reconstructed human epidermis to our extracts to look for irritation. Next, we score it, and we use an MTT version of the cytotox test to score. Basically, what that means is the more purple there, the less irritation. So we want to see purple. So in this particular picture, right, we see those purple wells. That means there has not been irritation there, where the clear wells had irritation. So we can actually determine how much irritation has been present by looking at this purple change. We also run it through interleukins, which is kind of a key signal for irritation. So we can actually see if there's been irritation present if we see clear cells. Last, we do a scoring criteria. So this is actually the scoring criteria. It's right there's the acceptance criteria, 70% viability. So if you're below 70%, then you're a possible irritant. If you're above 70%, then you're safe, okay? So here we have, you saw the turnaround times up front. We have a much quicker, cheaper, non-animal version of this test. So we should always run the in vitro irritation, right? Problem is, and we'll talk, is that the FDA right now does not accept the in vitro irritation. We're close. We've been working with them for 2.5, 3 years. We're close to getting them accepted. I would hope it's this year. There's a lot of data being produced throughout the industry. Medtronic just produced this great paper they presented at SOT last week that we're hoping to help convince the FDA. So we're getting close. But if you're doing MDR remediation, or if you're doing anywhere else in the world, this in vitro irritation will work, okay? It's only the FDA that doesn't accept it. So Japan, China, everywhere else, we've gotten a nod that they will accept the in vitro irritation. So don't just jump to the animal if you're going somewhere else or if you're doing this for some other reason other than submitting to the FDA. This test isn't the standard. It will be accepted by everybody else. And so it's a good option for you. The next one, besides, so we talked about physical chemical information. I do want to mention right here, the chemistry, like extractables and leachables, can take the place of all these arrows. So these are the ones that the FDA's agreed to use chemistry to replace. So as you can see, if you have to do material-mediated pyrogenicity or acute or subacute or chronic genotox, carcinogenicity, extractables and leachables with a toxicological risk assessment can take the place of those. And let me tell you, we're getting better and better at extractable leachables all the time. So a year ago, the FDA and the labs were having these back and forths and arguments, but we've been working with the FDA. We're getting better and better to the point where we're seeing less and less pushback from the FDA for extractables and leachables. So we should be able to do these tests for quicker and cheaper than any of these animal tests, especially today's market. So I do want to also mention here that if you have a device that is short-term contact, then the chemistry is not going to help us except for maybe some sensitization evaluation. It helps us with prolonged and especially permanent contact devices. So those are the ones we want to start using chemistry for. But now in this market, there are times we can start using chemistry to help us with sensitization evaluations. Usually we would tell you not to because sensitization is 8 to 9 weeks historically, and it's just safer to do the test. But now that the sensitization test has ballooned out to 18 weeks, we have been starting to use chemistry to help us evaluate sensitization. I would recommend working with a toxicologist to be able to identify your samples, look at potential sensitizers in your process, make sure the chemistry can screen for those possible sensitizers. But it is something to think about and work with your lab to be able to help identify and quantify any potential sensitizers so that you don't have to do that test. Okay, so those are kind of the tricks and tips for shortening your timelines right now in this market. I do want to just mention one last time, don't checkbox tests. And I know it was easy to do back in 2 years ago, but right now I hear so much frustration from my clients about turnaround time. They're like, you kidding me? The sensitization test is that long? Or my subacute subchronic's not going to be done until when? I can't really control the animal labs out there and their capacity. But what we can control is evaluating the biocompatibility of your devices and do a better job of evaluating instead of testing and get it on the market. And like I said, we've had lots of success doing that. So it's not like we've never done this before. So be creative, think about it, and in the end, let's help you out. Here's a couple of publications that I thought you guys would like. And by the way, for those of you just coming in new, if you want a copy of these slides, just bring me your business card. I'd be happy just to email you the whole deck so you don't have to write anything down. Thank you for your time. Are there any questions I can help you with? It's a great time to ask questions, guys. Yeah. His question was, is there going to be more of -17, -18 testing? So first off, -17 is in draft right now. So that's going to be a new standard coming out. -18 is being re-looked at today too. So we're changing those standards all the time. But with this impact that we're seeing, we've never seen chemical characterization be more of a demand today. So we see chemistry being able to basically replace any of the systemic toxicity endpoints, possibly even including sensitization, if done correctly. So what that means is that if you have anything that requires a systemic toxicity endpoint, chemistry is probably going to be your best bet to reduce your turnaround time and price. So yes, we see that becoming much, much more important in the future. In fact, the whole standard's kind of being rewritten to risk assessment and chemistry as the future of biocompatibility. I have Dr. Jorgensen's in the back. He's my chemist. He loves me saying that because historically I don't give chemistry enough credit. But today I would say that I think chemistry becomes more and more important for biocompatibility in the future. Good question. Thank you. Any other questions, guys? Yeah. Great question. So her question is, if your device has already been approved with the 510K, do you have to re-qualify or re-go through the standards? So with the 510K, how it works, if there's been no changes to that device, then you're fine. You can just keep on going through. You don't have to meet this current standard. But in today's market, with the supply chain we're seeing, all I'm hearing is that, oh, we can't get that material, or we can't get this material, we have to change materials. I'm seeing more changes today than I've ever seen in my career. So I would highly imagine that most devices out there have not changed even a little bit in the last few years. And if you have a change, you may have to reevaluate to the current standard. So that's when the gap analysis might come in, things like that. But if you're lucky enough to not have to change your device, then you're fine just keeping with what you did previously. Yes. So if I heard your question, sorry, if I heard your question right, like they may be changing it, but you're not. So they have to requalify to the current standard, but yours is not been, right? Yeah, so then they would have to go back and either ask you to do something new, or they would have to do it themselves, right? And so that's the decision they have to make as a device manufacturer. Good questions. Any other questions? Okay, make sure you come up and grab a fun slide rule because, those are free. And if you want a copy of the presentation, give me your business card. But thank you for your time. It was great talking to you guys.